Biotechnology is a revolutionary field. It harnesses the power of living organisms for practical applications. Imagine a world where we can create life-saving drugs, a world where crops can resist diseases. That's the promise of biotechnology. It's a fusion of biology and technology, and it's transforming our lives in remarkable ways. From medicine to agriculture, biotechnology is leaving its mark. It's helping us develop new therapies, it's improving food production, and it's even cleaning up our environment. This powerful tool has the potential to address some of humanity's biggest challenges. It's an exciting time to explore the world of biotechnology. One of the most important aspects of biotechnology is genetic engineering. So let's dive deeper into the fascinating world of biotechnology. Genetic engineering is like editing the blueprint of life. It allows scientists to manipulate DNA. This is the molecule that carries genetic instructions. By changing these instructions, we can alter the traits of an organism. Imagine correcting genetic defects or enhancing desirable characteristics. That's the power of genetic engineering. For centuries, humans have been modifying organisms. We've been doing it through selective breeding. Think of the different breeds of dogs. But genetic engineering is different. It's more precise. It allows us to introduce specific changes to an organism's DNA. This precision is a game changer. Genetic engineering offers several advantages over traditional methods. It's faster, it's more accurate, and it allows for more targeted modifications. This opens up a world of possibilities. To understand the magic of genetic engineering, we need to meet its tools. These are special enzymes that act like molecular scissors, glue, and copy machines. They allow scientists to cut, paste, and copy DNA fragments with incredible precision. It's like having a nanoscale toolkit for manipulating the building blocks of life. One of the key players is restriction enzymes. These enzymes are like molecular scissors. They cut DNA at specific sequences, creating fragments with known ends. These fragments can then be joined together with other DNA fragments. This is done using another enzyme called DNA ligase, the molecular glue. Another essential tool is DNA polymerase. This enzyme acts like a molecular copy machine. It replicates DNA strands, creating multiple copies of a specific DNA fragment. This is crucial for amplifying DNA samples. Vectors are like delivery vehicles. They carry foreign DNA into host cells. Recombinant DNA technology is at the heart of genetic engineering. It's like cutting and pasting different pieces of DNA. This creates new combinations of genetic material. Imagine taking a gene from one organism, then you insert it into another. This can give the recipient organism new traits. It's a powerful technique with far-reaching applications. The process begins with identifying the gene of interest. This gene might code for a desirable trait, like disease resistance in a crop or insulin production in bacteria. Once the gene is identified, it's isolated from the source organism. This is usually done using restriction enzymes. The isolated gene is then inserted into a vector. This vector acts as a vehicle to carry the gene into a host cell. The vector containing the gene of interest is then introduced into the host cell. The host cell becomes a factory for producing the desired protein. Section 5. A new home for genes. Introducing recombinant DNA into host cells. Imagine you've created a piece of recombinant DNA. It carries a gene for insulin production. Now, you need to get it inside a host cell. This cell will then become a tiny factory, churning out insulin. There are several methods for introducing recombinant DNA into host cells. One common method is transformation. Another method is electroporation. This creates temporary pores in the cell membrane. For animal cells, a common method is transfection. Special chemical carriers help the DNA cross the cell membrane. Viruses can also be used as vectors to deliver genetic material. Section 6. The factory of life, producing biotechnological products at scale. We've inserted a gene into a host cell. This cell can now produce the desired protein. But how do we scale up this process? How do we go from a single cell to a large-scale production line? That's where bioreactors come in. They are like giant fermentation tanks. They provide the ideal conditions for cells to grow and produce valuable products. Bioreactors are carefully controlled environments. They provide the optimal temperature, pH, and nutrients for cell growth. They also ensure adequate oxygen supply and waste removal. This allows cells to thrive and produce the desired product at high levels. Different types of bioreactors are used for different applications. Stirred tank bioreactors are common for producing proteins and enzymes. Airlift bioreactors are used for processes requiring high oxygen transfer rates. The choice of bioreactor depends on the specific requirements of the production process. Section 7. Biotechnology in Action, Real-World Applications and Innovations. Biotechnology is not just a futuristic concept. It's already transforming our lives in countless ways. From the food we eat to the medicine we take, biotechnology is leaving its mark on various aspects of our lives. It's a testament to the power of harnessing the tools of nature to address human needs. One area where biotechnology is making a significant impact is medicine. Recombinant DNA technology is used to produce a wide range of therapeutic proteins. This includes insulin for diabetes, growth hormone for growth hormone deficiency, and clotting factors for hemophilia. These treatments have revolutionized the management of these conditions, improving the lives of millions of people. Biotechnology is also playing a crucial role in developing new diagnostics and therapies for diseases. Gene therapy, for example, involves replacing or inactivating faulty genes. This has the potential to cure genetic diseases. The development of personalized medicine is another exciting frontier. This approach uses genetic information to tailor treatments to individual patients. Agriculture is another field that's benefiting from biotechnology. Section 8. The ethical landscape of biotechnology, weighing the benefits and risks. Biotechnology, with its immense power, comes with great responsibility. Ethical considerations are paramount. Open discussions about the benefits and risks are essential. Genetic engineering is a major ethical concern. Critics argue it's unnatural and worry about unintended consequences. Long-term effects on the environment and health are debated. Genetic information raises privacy concerns. Strong safeguards are crucial to prevent misuse. Despite concerns, biotechnology promises solutions to pressing challenges. Section 9. Biotechnology and the Future. A glimpse into the possibilities. The future of biotechnology is brimming with exciting possibilities. Imagine a world where diseases are diagnosed and treated before symptoms even appear. A world where crops are engineered to thrive in even the harshest conditions, ensuring food security for all. A world where personalized medicine tailors treatments to individual patients, maximizing efficacy and minimizing side effects. Nanobiotechnology is an emerging field that combines the power of biotechnology with nanotechnology. This involves using nanoscale materials for biological applications. Imagine tiny robots delivering drugs directly to cancer cells or nanosensors detecting diseases at their earliest stages. The potential applications of nanobiotechnology are vast and hold immense promise for medicine, agriculture, and environmental science. Synthetic biology is another groundbreaking field that aims to design and create new biological systems. This involves using engineering principles to build biological parts, devices, and systems. Imagine creating artificial cells that produce biofuels or engineer bacteria that clean up oil spills. Synthetic biology has the potential to revolutionize various industries. Section 10. Conclusion. Embracing the promise of biotechnology. Biotechnology has emerged as a transformative force. It's reshaping our world in profound ways. From revolutionizing medicine to transforming agriculture, biotechnology offers solutions to some of humanity's greatest challenges. As we continue to unlock the secrets of life, the potential applications of biotechnology are limitless. The journey into the world of biotechnology is filled with wonder and excitement. It's a journey of discovery, innovation, and pushing the boundaries of what's possible. The future of biotechnology is bright, filled with possibilities that can improve lives and create a more sustainable future. Section 11. Glossary of Terms. Biotechnology. The use of living organisms or their components to create products or processes for human benefit. Genetic engineering. The direct manipulation of an organism's DNA to alter its characteristics. Recombinant DNA technology. Techniques used to create DNA molecules by combining DNA from different sources. Restriction enzymes. Enzymes that cut DNA at specific sequences. DNA ligase. An enzyme that joins DNA fragments together. DNA polymerase. An enzyme that replicates DNA strands. Vectors. DNA molecules used to carry foreign DNA into host cells. Host cells. cells used to produce biotechnological products from recombinant DNA. Bioreactors, vessels used for large-scale production of biotechnological products.